So in this video, we'll be discussing about glycolysis pathway. Glycolysis pathway takes place in the cytosol of the cell. What do you mean by glycolysis? Glyco means glucose, lysis means breakdown. Simply we can say that glycolysis is nothing but breakdown of glucose. It involves a series of reaction converting glucose to the pyruvate with the production of ATP. Firstly, glycolysis has three phases: energy investment phase, splitting phase, energy generation phase. Glucose gets converted to glucose 6-phosphate in the presence of an enzyme hexokinase or glucokinase. Simply we can write glucose and here one phosphate group is going to add, so we have to write kinase, glucokinase, as it is 6-carbon compound hexokinase, as phosphate group is going to add. So in the presence of ATP is converted to ADP and inorganic phosphate. This phosphate group adds to this glucose and forms glucose 6-phosphate. Glucose 6-phosphate reversibly converts to fructose 6-phosphate in the presence of an enzyme phosphohexoisomerase. Here the reaction is isomerization, no? So we have to write isomerase. Phosphohexo, as both are 6-carbon compounds, hexoisomerase, phosphohexoisomerase. Next, fructose 6-phosphate is converted to fructose 1,6-bisphosphate, phosphofructokinase. As I said, here one phosphate is going to be added. ATP is converted to ADP plus, and the inorganic phosphate gets added to this fructose and fructose 1,6-bisphosphate is formed. Till this, it is energy investment phase. Here the ATPs, you are investing the ATP as the energy source. Splitting phase. This fructose 1,6-bisphosphate is a 6-carbon compound in the presence of an enzyme aldolase. It splits into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These both are 3-carbon compounds. These are interchangeable by these two are interchangeable by the enzyme phospho, as both contain phosphate group. Triose is a 3-carbon compounds. Isomerase, these are interconvertible isomeric forms. Phosphotriose isomerase. So one molecule of glucose produces 2 molecules of glyceraldehyde 3-phosphate. From this, energy generation phase start. Glyceraldehyde 3-phosphate converts to 1,3-bisphosphoglycerate by producing NADH in the presence of an enzyme glyceraldehyde 3-phosphate dehydrogenase. So NADH is being produced. Now we have to write dehydrogenase enzyme and the same substrate name is added. So glyceraldehyde 3-phosphate dehydrogenase. 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. In the here the 1-phosphate group is added to ADP and here ATP gets synthesized. So phosphoglycero as substrate, kinase. So ATP as phosphate is involved, we have to write kinase, phosphoglycerokinase. 3-phosphoglycerate is converted to 2-phosphoglycerate. Here nothing but the phosphate group position gets changed from 3rd position to 2nd position. Same we have to write substrate and mutase. The positions of phosphate is changed. Phosphoglyceromutase enzyme. So 2-phosphoglycerate by removal of one water molecule in the presence of enolase enzyme forms phosphoenol pyruvate. This phosphoenol pyruvate in the presence of pyruvate kinase. Same pyruvate kinase. Here we write kinase means one ATP molecule gets synthesized. So pyruvate. This pyruvate is the end product of glycolysis in aerobic pathway. And this pyruvate again converts to lactate in the presence of lactate dehydrogenase and NADH gets synthesized. This occurs in anaerobic pathway.